All right, so we just talked about semiconservative replication. So let's look at this a little bit more in depth and see how this occurs. So we're starting off with our original DNA molecule that's wrapped in this double helix. And we have a couple of enzymes that are going to help with this process of replicating or copying the DNA. The first molecule, or the first enzyme I should say, is helicase. We recognize that helicase is an enzyme because it has that ASE ending to it, and the enzyme always is named after what it does. So this is an enzyme that's going to work on the helical structure of the DNA molecule. So it's going to unwind the DNA and flatten it out so that it's no longer wrapped in that spiral shape. It will also break the hydrogen bonds between the nitrogenous bases. So we see here our helicase on the left is kind of joined together, our two enzymes, and then on the right we see that the two enzymes are moving apart. One's moving towards the top and the other one's moving towards the bottom. And as they move apart, they're unwinding the DNA and they're breaking the bonds between the nitrogenous bases so that they're exposed. So DNA helicase, once again, works on the helix and breaks the bonds so that we have our two template strands. After that, we have another enzyme that's called DNA polymerase. Once again, ASA ending, so you know it's an enzyme, and it's a polymerase molecule, so it's going to make a polymer. So what polymer is it going to make? It's going to make DNA. So DNA polymerase is responsible for adding new nucleotides to our template strands. So it's going to build the complementary template strand to our old DNA molecule. So in dark blue, you see DNA polymerase here. DNA polymerase needs a little help to get going. So in red, there's what's called a primer. So the primer gives it kind of an end to attach to start attaching nucleotides to. So as DNA polymerase moves along the old strand here on the bottom, it's going to look at which nitrogenous bases are exposed. So if we're looking at this top strand here and saying this is our old nucleotide strand, if it sees an adenine at the top, DNA polymerase will bring in a nucleotide that has a thymine and hydrogen bond it to the adenine. If there's a cytosine on the old nucleotide strand, DNA polymerase will bring in a guanine to match up against it, and it will bond together. So same thing, you can kind of go down the strand and see once again that G is equal to C and T is equal to A, and as we go down. So if there's an adenine that's exposed here on the old strand, DNA polymerase would bring in a thymine. If there's a guanine that is exposed on the top strand, or excuse me, cytosine that's exposed on the top strand, then there would be a guanine that would be put into position by DNA polymerase. So DNA polymerase keeps on doing this. So you'll have a polymerase that is attached to the both nucleotide strands. So what we were looking at before, you would have one on the top and then one on the bottom. And so in this image, you can see the old DNA molecule, which is right here. So what's happening is replication's proceeding in this direction. Oops, got a little too close there. All right. And helicase is moving from right to left, and it's breaking the bonds in the middle here and flattening out the DNA molecule. And then polymerase has been moving along and building a complementary strand, and it will continue to match up strands. So if there's an adenine here, it would put a thymine across from it. There's a cytosine there, right? It would put a guanine across from it. All right, sorry, my drawing's not the greatest there, but it's going to continue to build a complementary strand. And then you see it winding back up again and forming two strands there. All right. Remember, each strand consists of one old nucleotide strand and one new one. And that's the case for both the top and the bottom. Now let's talk about a similar process that is used in the lab. So what we've been talking about with semi-conservative replication is what occurs naturally within the cells, and we said this is a precursor to cell division. And we are able to kind of capitalize on that process to be able to make copies of just portions of the DNA molecule. So this is what we call the polymerase chain reaction, or PCR. So this is a lab technique. So this is something that is performed in the lab to make lots of copies of just a portion of the DNA molecule. So if there is a region of the DNA that is kind of desirable that you wish to look at, or you wish to conduct research on, or you want to sequence that portion of the DNA and know what the nitrogenous base sequences of it, you can make billions of copies of that segment using this process. So down in the lower left-hand side, we've got a little PCR tube. It's not to scale there, but it's showing you that this is where the reactions will take place. So we have the name polymerase. So that's referring to the fact that we need a polymerase enzyme. And we talked about DNA polymerase for semi-conservative replication, but we use a polymerase enzyme as well for the PCR reaction because we need that enzyme to help build our new DNA molecule. So within that little PCR tube, we place our DNA, our sample DNA that we wish to make a copy of. We've got some buffers in there. We've got a primer. Remember, we talked about the primers as just a short segment that helps polymerase start to attach nucleotides to. And these DNTPs refer to the fact that these are just our extra nucleotides. So you need extra adenine, thymines, guanines, cytosines so that you can build these new DNA segments. So over on the right, they're showing an example here of the DNA template, and just in red is the section that's going to be copied. So once again, we're not copying the entire DNA molecule, we're only copying just that little portion right there. So that's one of the differences between semi-conservative replication and the PCR reaction. So another difference is the fact that we're not going to rely so much on the enzymes. We're going to use temperatures instead to help with this process. So instead of having a helicase enzyme, we're simply going to use a hot temperature to help us, to help the DNA molecule lose its shape. So this polymerase chain reaction is called a chain reaction because we go through several cycles. And each cycle consists of a denaturation stage, an annealing stage, and an extension stage. So denature refers to the fact that the molecule is kind of falling apart. It's losing its shape. So it's going to lose that helical shape, and the hydrogen bonds between the nitrogenous bases are going to break apart. So we really don't need the DNA helicase enzyme. We can simply subject our molecule to a higher temperature so that it falls apart. The annealing stage is when our primers attach and DNA polymerase attaches to those primers, followed by the extension stage where DNA polymerase starts to build our complementary strands. So that is just one cycle. and we can go through this cycle about 30 times. And just in 30 cycles, we will have billions of copies of that targeted region of the DNA molecule. So this can be used for a wide variety of different purposes. A lot of times it is the first step for further research. So it can be used for paternity testings if you're trying to detect mutations in the DNA molecules. If you're doing some type of recombination of combining two DNA from two different sources, PCR would be your first step for all of those. And the last technology that I want to talk about is CRISPR. So CRISPR-Cas9 is more recent than the PCR reaction, and it can get quite involved, so I don't want you to get too bogged down in what is going on here. But CRISPR-Cas9 basically consists of RNA molecules and proteins, which you're seeing on the top right there. We've got two of them, and below that is the DNA that they're going to be targeting. And in green, let's say that is the region that we are looking at. That's the region that we're interested in. So these molecules can come in and target either end of that region of the DNA and basically cut it out. So the molecules that are part of, you've got the RNA guide molecules that kind of help the Cas9 to detect the region that it wants to target or it wants to cut. And it will cut either end of it, and therefore you see all these slashed lines through that region, noting that that is the region that is going to be removed. And then once that area is cut out, you see the two ends coming together again. So DNA can repair itself after this process has happened. So CRISPR-Cas9 is one way in which a section or a portion of DNA can be removed. And then from there, other procedures can take place to insert new gene segments or do other type of techniques.